Thank you very much. It is my great pleasure to present you a small piece, but very potent piece of our research. So let me introduce you the hunt for Lloviu virus in Europe. This is Lloviu because it is a Spanish word. So if some of you learned some Spanish, maybe you will get some idea about this virus. And by the way, this is the picture of this virus, and we will speak about this picture. As you can see, it is like a filament, and this virus will be a filovirus because how it looks like. Most probably all of you know about Ebola because everybody knows about Ebola. Virologists like me, we learned a lot about Ebola since the West African Ebola outbreak, and many of you, most probably many of you, already saw the TV show of Hudson or maybe read the book, the Hudson book. And we had quite huge challenges with this virus, and still we have many challenges with this virus, and not just with this virus, but the whole family of these viruses. And we call the family of these viruses filoviruses. I used to say that we used to speak a lot about the age of pandemics, which we live in now. And for sure, filoviruses have a huge chapter in the age of pandemics. If you check the news from like the last 4 weeks or let's say 2 years, you will meet a lot news about Marburg virus outbreak, recurring Ebola outbreaks in Congo, never-ending story. This is truly a never-ending story, and this is just from like last week. We have another Marburg virus outbreak in a totally new place, another country again. So this is like happening all the time with increasing intensity and frequency. It's not surprising. If you check the situation in Africa, you will find that the population boom in Africa is like the most potent compared to all the other regions of the world. And in parallel to this, we are cutting down the rainforests, we are disturbing these ecosystems. So we will more and more frequently meet with these viruses, and we will establish more and more connections with these viruses. So that is the main reason of these more and more frequent Ebola, Marburg, and other filovirus outbreaks. Bad news, it's not only in Africa. Since the huge developments in molecular biology, we were able to identify, not just we, but all of our colleagues and many other virologists in the world, we were able to identify many new species within the filovirus group. And the problem with these viruses, most probably some of you already have the question, okay, if we have these filoviruses here, what's the situation with these viruses? Can these viruses infect us or our animals like Ebola or Marburg virus? How they behave in the nature? What is the source of these viruses? How can we catch them? How can we interact with these viruses? Can we do anything to prevent the outbreaks, the spillover events? These are the most important questions. And these are selfish questions, of course, because we are humans, so we care mostly about ourselves. But you will see there are a lot of other questions connected to these. So, as I told you, the most important question and first question is finding the source of these viruses. If you identify a virus in an animal, that's not for sure it is the source. It can be like a result of a spillover event from an animal to another animal. The source, we call the source, there is a word reservoir. It means that somewhere in the nature there is a nice, like a long established collaboration between a virus and an animal. It's an evolutionary relationship, and usually these animals, reservoir animals, are doing fine with these viruses. Usually they are asymptomatic, they are spreading the virus without any problems. And bats are very special in this regard, as you may have learned during the last 3 years. Bats are very special in many ways, but they are reservoirs of many, many viruses. In case of filoviruses, we know, like, after 60 years of research, that Marburg virus has the reservoir of Marburg virus are those cute bats, so-called Rosetus aegyptiacus, but this is just, you see it's 60 years of research and still just a few results. So we can only say in case of Marburg virus that, okay, people, for sure, we know the source of this virus, so please try to lower the connection with these bats and try to save the habitat, the natural habitat of these bats. So at this point, let me introduce you the European Ebola virus. It was discovered in relation to an event which affected the whole Iberian Peninsula in 2002, and it took nearly 10 years for the researchers to find the causative agent of these die-off events. And this was a new member of the filovirus genus. This was very frightening at that time, and most of the filovirus researchers were shocked because, oh my God, we have a filovirus in Europe, so what to do? Of course, we did what every filovirus researchers do. We started to look after the source. We started to describe those questions or answer those questions which I already presented you. So here is one specimen from Hungary from 2016. And as you can see, this poor bat presents some very similar symptoms like which we know from humans. So Ebola virus and Marburg virus, sometimes they have a manifestation, so-called hemorrhagic fever. So some people present hemorrhagic symptoms, and very interestingly, these bats sometimes, like in 50% of the cases, they present something similar. So this is a very strange virus. And because it's a very strange virus, I have to present you the hypothetic source of the virus. This is the Myneopterus bats. They are very special bats in Europe. We have 32 bat species in Europe, and this is the only bat species which forms huge colonies, like tens of thousands of bats at one place. And as you can see, the distribution range of this bat, it covers the whole Mediterranean region and some more regions actually to West Asia. So it has a huge distribution range. And if we couple this bat with the virus, of course, we have several questions on the whole region. So how our work began? We began our work in 2013 when we recognized that 5% of the Hungarian Myneopterus bat population just perished. This was very strange at that time, and many bat researchers get in touch with us and asked us to verify the causative agent or what is happening in Hungary. We were not lucky enough to obtain fresh samples. For virology, you always need very fresh samples. So we had to wait for some more years, and in 2016 we were lucky enough to find some fresh bat specimens, and we were able to identify the virus in these bats. So actually our work began in 2016, but the whole, let's say, hunting, we call it virus hunting because actually it's very similar to hunting, it began earlier. So the research questions, the most important research questions, and the first questions you ask is, where can we find the virus? Of course, we knew that it's related to Myneopterus bats somehow, but where can we detect the virus? So if we can answer where can we detect, like in blood, urine, ectoparasites, whatever, we may have some ideas about the possible spillover events. Let's say, how can you catch it if you touch this bat? Is there a season for the virus? That's another important question. When should we close the caves and do not let anybody in to prevent the spillover events? What is hidden in the genome of the virus? The genome, as some of you, if you follow the SARS-CoV-2 pandemic, you may read about some very nice analysis about virus genomes, and actually the genome of a virus can tell you a lot of things about let's say, the behavior of the virus. So this is a very interesting question. And most importantly, and this will be the biggest achievement of our work, which I present you now, how can we use this virus in the lab? Because if we can get it in the lab and we can culture the virus, let's say, we have 10 or 20 more years of research with this virus. So before starting, we had to jump over a lot of challenges. So all of the bat species in Europe are highly protected. So we had to develop our tools and techniques. How can we examine the virus without killing these bats, without invasively sampling these bats, without disturbing these bats? So we had to develop a lot of techniques, from modern genomic sequencing to PCR tests. You are all familiar with PCR tests, to serology tests specifically for these bats. And we had to put all these capacities into a mobile lab because these bats are living very far from humans, so we can reach them very, very hardly. So there are a lot of challenges to go there. So it's very hard to obtain the fresh samples and then get back fast to the lab. So it was more practical to bring the lab to the cave and do all the analyses there. And we have some very nice collaborators in the Hungarian army medical research center, and we stole some ideas from them and we built our own mobile lab. Actually, we used this mobile lab recently in Bangladesh, so this is a very good capacity to examine viruses locally and for outbreak investigation, outbreak prevention, so things like this. But in this case, in this research, it was very useful to step forward. So let me show you briefly, of course, the field virology results. We were able, as I told you, to identify the serostatus of these bats. You are, I think now after COVID, sorry for this, but you are all familiar with antibodies. So after COVID, you can check your antibodies. Did I have the disease at some point or not? We can do the same with these bats in relation to this virus. So I can tell you that were these bats infected, how many of these bats were infected, and we are able to check the PCR status of these bats and all the related ectoparasites and all the other related samples. And if you check this tiny grilled chicken here, it's not a grilled chicken, it's an ectoparasite. It's a very special ectoparasite of bats. It's called the bat fly. They have 60 million years of coevolution together. They have a very strong coevolution, so these are flies without wings because they just need the bats, so they live totally together. And what was a huge step, a huge achievement of this study is we were able to identify the virus in these bat flies, so we are digging deep now into the role of these bat flies in the coevolution of viruses and bats. They are very, very interesting. It's a more closer look to this tiny bastard, but yes, so they are very interesting for virologists now, and this is a very hot topic in virology to understand how viruses go through bat populations, how they are transmitted between bats, and we suspect that these animals may have a serious, significant role. Of course, the question, I already represented you the geographic range of these bats, so we were very interested in other places. So in every summer, I spend just very few days at home, and we spend most of our time going across Europe and also Algeria and many nice places to check for the presence of this virus, to check for the serostatus of this virus and all the other things. So this is a very important question to solve. Where else you have to be careful with this virus, or you have to take care of not getting infected. Let's see the laboratory results, and this is the most significant result of our current work. Isolating a virus, it means you let's say culture the virus in a flask, and this is, I used to say, this is the holy grail of virologists because this opens up a lot of possibilities. What possibilities? Like developing antiviral drugs, developing vaccines, answering cell-level questions like what is the pathomechanism, what happens in the infected cell when this virus is there. So a lot of possibilities for our next 10 or 20 years. And this was a very challenging thing to do because we tried it for years. And then finally we had to obtain a very special cell line from Japanese colleagues from a similar bat species. It's also a Minopterus bat, but another species. And we had to use its, let's say, own cell line to finally have the success and establish the isolates of the virus. Actually, now we have multiple isolates from different regions in Europe. So this is a very important thing to do. This is, for me, this is the most interesting question at this point. Bats can teach us a lot of, bats can answer a lot of things which we need now. So how can we tackle pandemics? How can we face the next pandemic? Because as I told you, bats can easily live with viruses without symptoms, without getting the disease. And this is a very important question now. This is from Nature News feature. So they can teach us a lot of things. So now we are digging deep into these hemorrhagic symptom bats, and we try to figure out what is happening on the cellular level, what is happening inside these cells, inside specific infected cells, what happened there, what did the virus in these cells. So our future directions are following the same questions. So is it a reservoir? I mean, Minneopterus bats are really reservoirs. And if they are reservoirs, why 50% of the bats die with hemorrhagic symptoms of the virus? So this is a very interesting thing to answer. Is it pathogenic to humans? So now we were able to say that on the laboratory level, we were able to prove that actually this virus can infect human cells, which sounds very bad, but infecting human cells in the lab, it's very far from infecting the whole body, infecting somebody. So we have to solve, to answer this question, is it pathogenic? We have to solve a lot of questions. And we have to solve a lot of questions about the ecology of the virus, the seasonality, how can they transmit it to humans or other animals, so things like this, which I already presented you. And my favorite part is the viral genomics, the evolution of the virus. Actually, the viral genomic research can tell you even the source, I mean the evolutionary source of the virus, so the evolutionary story of the virus, and this is very interesting. So in summary, we have a filovirus in our backyard, which is like, sounds bad, but we are very lucky because we have this filovirus. Maybe in the near future, we will be able to solve those questions which we have now for 60 years with those African filoviruses. So maybe using this virus, we were able to solve those questions which cannot be solved in Africa, because doing research in Africa, it's very challenging. Putting the lab there, it's very challenging. Putting resources there is very challenging, but we have this virus in the backyard. So we are, in this term, we are very lucky to have the opportunity to make research with the actual filovirus. So we think that this will be a perfect model for other filoviruses as well. And this is where we are. This is another aspect of science. We are very proud that this was a significant finding, because for 60 years there was no step forward with filovirus research, and finally we were lucky enough to isolate. And this is a piece. This is a highlight in Science Journal, and also we were invited to a nice podcast. If any of you want to be a virologist, please follow the This Week in Virology podcast. I think there is no virologist in the world who does not follow this. And of course, as I told you yesterday, and as you may have seen in the previous presentation, science is teamwork. So I want to say thank you for all of the team members, and especially for the, let's say, most brave team members, the field team. And special thank you for your kind attention. I hope you enjoyed this presentation. Thank you very much. Thank you.